Okay, so we left off going 100 miles an hour, trying, or at least I was going 100 miles an hour, trying to get through this ONZ, OMP-R two-component system. So we left off having explained the fact that OMP-F and OMP-C are the two outer membrane proteins that dictate whether E. coli can survive optimally in either a freshwater environment or in the colon. So if the bacterium wants to survive in a water environment, an aqueous environment, so low solute concentration, it is best for it to express OMP-F. And it's best for it to express OMP-C in an environment like the colon, where solute concentrations are very high. So the way that this is accomplished is by this ONZ histidine kinase protein sensing what we believe to be membrane turgor. So as solute concentrations are low, the turgor pressure of the cell changes. And so what you're seeing here is that this artist has represented the conformation of the signal input domain of ONZ as being somewhat collapsed under high solute concentration, as if somehow by osmolarity that protein has been collapsed. And then when solute concentrations are low, as would be the case in a freshwater environment, ONZ has this alternative conformation. And so in that low solute concentration, reflective of E. coli being in a water environment, the kinase activity of ONZ is minimized. Not eliminated, not stopped, but minimized. So when that occurs, the amount of phosphate groups that are being transferred to the response regulator, OMP-R, are minimum. So this form, the OMP-R phosphate, is going to be at very low levels. OMP-R non-phosphorylated is going to be at high levels. Now, let's look at the loci for the two genes that encode OMP-F and OMP-C. Let's start with OMP-F. We've started with a situation where E. coli is in a water-type environment, a dilute environment. And so OMP-R phosphate is going to be at low concentration. So if you look at the two sites upstream of the OMP-F gene to which OMP-R phosphate can bind, it has its choice of a high affinity site, shown here as labeled as on, and a low affinity site, labeled as off. Now, when OMP-R phosphate is at very low levels, in essence, the only place that OMP-R phosphate is ever going to bind is going to be to the high affinity site, which is an activator. So this particular site tends to cause RNA polymerase to bind and to transcribe OMP-F. So in that environment, in that high water environment, you tend to get transcript of OMP-F. You don't, on the other hand, when OMP-R phosphate is at low concentrations, tend to get transcription of OMP-C. What's shown here is that there's a single site where OMP-R phosphate can bind at this locus. So we'll talk about MYC-F in a minute, but OMP-R phosphate, if it binds to this site, will turn on the transcription of OMP-C and MYC-F. But notice the fact that the on site here is low affinity. So under freshwater conditions, when OMP-R phosphate is at low levels, it will bind only very rarely, if ever at all, to this low affinity site to turn on OMP-C. So for the most part, in that freshwater environment, it's going to bind to this high affinity site and no place else. It's going to turn on OMP-F. OMP-F is going to be transcribed into this messenger RNA, which will then be translated to produce this protein OMP-F. And E. coli lives quite happily in that water environment for a while. Now, what happens when E. coli gains access to a new host. Some animal drinks some contaminated water, or perhaps even you going for a swim in the Crimdell, which you shouldn't do, or in Lake Matoka, which you also shouldn't do. But if you did and consumed by accident a little bit of water, you may get some E. coli in there. And so the E. coli has been surviving in the water because it's expressing OMP-F and not OMP-C. But now it gains access to your intestinal tract where solute concentrations are higher. And so that signal, those increased osmolarity, is detected by the membrane histidine kinase ONZ. And so now the situation is such as shown here on the right-hand side. ONZ, because of the high turgor pressure, because of the high solute concentration, now tends to have its kinase activity maximized. So now the phosphoryl group is being put onto a histidine on this kinase and being transferred again to OMP-R phosphate. But now OMP-R phosphate is going to be the more abundant form of OMP-R, much more so than OMP-R unphosphorylated. So now if we come over here and look at how OMP-F and OMP-C are going to be affected in this condition, where OMP-R phosphate is present at high levels, first let's deal with how the bacterium begins to express OMP-C, shown at the bottom. OMP-R phosphate, now at high concentration, is actually going to bind significantly to this site controlling the transcription of OMP-C. Even though this is a low affinity site, because OMP-R phosphate is now at a high concentration, there is an increased chance that OMP-R phosphate binds to this ON site, even though it's low affinity, to turn on the transcription of OMP-C. That messenger RNA will now be translated into the protein OMP-C. Now, the bacterium also needs to be able to stop the production of OMP-F at the same time. If the bacterium is expressing OMP-C and OMP-F, this is not adaptive because you still have the high ion flux OMP-F, potentially allowing in toxic molecules in the intestinal tract through this large pore. So the bacterium needs to simultaneously increase the transcription of OMP-C and simultaneously stop the transcription of OMP-F. And so that's what you see with this second site on the OMP-F promoter region. This off site is a repressor site. This is a site that if OMP-R phosphate binds to, will stop RNA polymerase from transcribing OMP-F, so this piece of messenger RNA will no longer be synthesized. So the same conditions that increase or really initiate the transcription of OMP-C also simultaneously turn off the transcription of OMP-F. Makes sense. The bacterium in this condition in the colon does not want to express OMP-F. But it's not enough at this point. It's not enough to just stop the transcription of OMP-F. There is all of this transcript which was pre-existing from inside of that cell from the time when the bacterium was existing in a freshwater condition. All of that messenger RNA still remains for several minutes, and so it can be translated into OMP-F, and so many proteins, copies of OMP-F can be created. And so you have a situation where you have the potential to express both OMP-F and OMP-C at the same time, and that is never adaptive for any condition. E. coli wants to always maximize OMP-F while minimizing OMP-C, or maximize OMP-C while minimizing OMP-F. In the colon situation, where we are now, the bacterium wants to maximize OMP-C and minimize OMP-F. This is where this other gene down here, MYC-F, comes in. The same conditions and at the same time that this OMP-R phosphate predominates, and remember, OMP-R phosphate is going to predominate when the cell is in a high solute concentration, so that would be the colon environment. That high level of OMP-R phosphate binding to this one site not only turns on the transcription of OMP-C, it also turns on the transcription of this gene going in the opposite direction, called MYC-F. MYC-F is a gene that does not encode a protein. This is a non-protein encoding gene. It's an antisense or regulatory piece of RNA. MYC-F is only about 170 nucleotides in length. It has no Shine-Dalgarno sequence, so it is never bound by a ribosome. It is never translated into a protein product. What this MYC-F messenger RNA does under these conditions is that it has complementarity to this site labeled as off on the OMP-F messenger RNA. That's a mouthful. So this MYC-F messenger RNA is anti-complementary to a sequence of DNA, a sequence of RNA on the messenger RNA for OMP-F, and so it will bind to it. What that does is that prevents the pre-existing OMP-F messenger RNA from being translated. So what this does for the bacterial cell is that it very rapidly stops the transcription of OMP-F and very shortly thereafter stops the translation of any messenger RNA that may still be remaining. MYC-F will bind to this end of the messenger RNA and prevent ribosomes from binding. Remember, ribosomes can only bind to a Shine-Dalgarno sequence at the single-stranded RNA level. If that Shine-Dalgarno sequence is occluded by RNA, if it's in a double-stranded situation, such as we saw in the ribose switch. If you look back at the slide on the ribose switch, you'll see that ribosomes could be occluded. They could be prevented from binding by a particular secondary structure, stem loop structure. In this case, the occlusion is due to a distinct piece of RNA, and that's what's shown here. So on the bottom here, shown in orange, is the outer membrane protein F messenger RNA. So this is the mRNA that's been hanging around for a few minutes. Bacterial mRNA has a relatively short half-life, but it's been hanging around for a few minutes since the bacterium found its way into the colon. And so if it's there, it has the capability of binding a ribosome, being translated into a protein that the bacterium doesn't need. That condition of going into the colon turns the ONZ protein on, which leads to increased phosphorylation of OMP-R, which leads to an expression of MYC-F. And now you can see that the MYC-F is antisense to the OMP-F. Here's the 5' end on the right of the MYC-F messenger RNA, and you can see the base pairing. So it's not a perfect match, but it's enough so that ribosomes can't find their way onto this OMP-F messenger RNA. So the system is very elegant in its ability to simultaneously stop transcription of OMP-F and at the same time, or nearly at the same time, to prevent the translation of transcripts that are already there. So we can look at this in another way. So we can look at the expression of these proteins as a function of osmolarity. So as osmolarity is increasing, as we look at this from left to right, the OMP-R phosphate levels, that response regulator, are going to rise as the cell encounters higher and higher osmolarity conditions. So ONZ is become active, it's phosphorylated OMP-R, so the pool is shifting to OMP-R phosphate with time. If you look at the expression of OMP-F during the same gradation of osmolarity, you'll see that at low osmolarity, OMP-F is expressed maximally. And then, as OMP-R phosphate is increased due to changes in osmolarity, OMP-F transcription collapses, and it's largely zero at high osmolarity. On the other hand, if you look at OMP-C, you can see that OMP-C is expressed at lowest levels when OMP-F is at highest levels. And as osmolarity increases, so does the expression of OMP-C. So you can see that these proteins are largely mutually exclusive. For the most part, in E. coli, it produces one of these porins and not the other. You very seldom find a situation where you can find appreciable amounts of both of these porins at the same time. This is another way to look at that regulatory system. So what's being shown here in blue is the promoter sequence for OMP-F. That's what the F's here stand for. And at the bottom is OMP-C. The C's here standing for OMP-C. So these are the high affinity activator sites. So when OMP-R phosphate is at low levels, it can bind to these high affinity sites very well. The other sites, represented as F4 and as the C1 sites, are low affinity. So under low osmolarity conditions, any OMP-R phosphate that is generated tends to get sucked up in these high affinity sites, which promote, here's RNA polymerase, which promotes the transcription of OMP-F. So this wavy line is meant to represent the fact that under low osmolarity conditions, you get largely only the transcription of OMP-F. Now, as the cell finds its way into the high osmolarity condition, where OMP-R phosphate is at much higher levels, it still does bind to these high affinity these high affinity ON sites for OMP-F. But now it also binds to a low affinity site shown here as F4. When OMP-R binds to F4, there is a change in the DNA topology such that there's a looping here. The most important thing to remember about this is that this particular arrangement stops the transcription. So you'll see that RNA polymerase is not binding here. You'll see that there is a lack of transcription when this low affinity F4 site is engaged. At the same time, the low affinity sites in front of OMP-C are in fact engaged. So now these are the sites that activate OMP-C transcription. And they do this only at high solute concentrations because these low affinity sites bind OMP-R phosphate only when it's at very high levels, and then we get the transcription of OMP-C. Okay, that's not anything that we need to be concerned with. That's from another year. We won't go over that this year. So yeah, this is kind of another alphabet soup. If you go through this carefully, if it doesn't already make sense to you, which it might, but I just listen to the words coming out of my mouth, and so I can certainly understand why it might not be clear yet. But as you carefully go through this, and then as you come and talk with me at office hours and during review sessions, it will become clear, and suddenly it will all make sense, I promise. Okay, that's it for now. This does end the material for the exam next week. I will post a new slide set on Blackboard, hopefully tonight. Okay, thanks a lot.